Hi, everyone. Welcome to the CMC and Regulatory Alignment, the Ask Me Anything series that we're starting here at Syner-G about lessons from the front lines. I'm going to give it a couple more minutes to let a few people join, and then I will jump right into the presentation and the conversation. Okay, so I think we have a critical mass. So my name is Ray Forsland. I'm going to moderate this session today. My background is I'm Executive Vice President of Product Development here at Syner-G. I've been here a little under 10 years. I'm a PhD organic chemist, have started my career at Vertex, worked at Ironwood, had my own company for a little while, and then I've been leading the scientific consulting team for the past 10 years. I'm going to hand off to Stacy, who will introduce herself.
Yeah, hi everyone. My name is Stacy Plum. I'm a Senior VP of Pharmaceutical Development here at Syner-G. I've been with Syner-G for about 5 years now, and prior to that, I've spent about 25 years in the development of drug realm in terms of biologics. With regard to background, I have a PhD in microbiology and immunology, and have spent most of my career working for smaller startup biotech companies and leading them through all stages of the development process. I'll turn it over to Drew.
Awesome. Yeah, thank you. Drew Barlow. I've been with Syner-G Biopharma Group for about 10 and a half years now. I lead our regulatory teams within Syner-G, specifically 25 years in regulatory and regulatory CMC. I have a master's in public health and was a member of the United States Public Health Service, where I worked for FDA Office of Regulatory Affairs for about 5 years, which is now the Office of Inspections and Investigations. Then moved over to Vertex and Alkermes, supporting a number of preclinical, early development through approval and post-approval programs, mainly in the small molecule space and more recently into the large molecule space. So, and I'll turn it over to Ed, but thanks.
Yeah, thank you, Drew. My name is Ed Ciolkowski. I am Vice President of our small molecule pharmaceutical development group. I've been in the industry for a little over 30 years, trained as an analytical chemist, and spent most of my career in mid to large size pharma, including Upjohn, Pharmacia, Pfizer, and Bausch & Lomb. I've supported small molecule product development throughout my entire career, both in the vet med and human health space, and supporting drug substance scale-up, analytical testing, and drug product development all the way from first in human through post-approval changes. A lot of experience with solid oral dosage forms and a significant amount of sterile products and topical experience too. So I think that is, I've been with Syner-G for about 12 years. So Ray, I'll hand it back to you and let you go through the prepared materials.
Yeah, now that you know who we are, I just want to kind of lay out kind of what to expect from us in the next hour or so. So, the format, we want this to be really interactive. So within the Teams and the chat, use the Q&A function. As the moderator, I'll be monitoring that, and I can put questions directly to the panelists. So if there's something specific that you want to discuss or have them talk or follow up on, please use that function. We'll have all of this information available later. There's really not a lot there. This is more meant to be an open discussion. However, we are recording this, so the transcript and all of that will be available on the Syner-G website. And if anything comes up after the fact or you have any questions, feel free to email me, and I will either answer your question or put you in contact with the right people to be able to answer your questions. So without further ado, let's kind of begin the seminar. So, first off, I know there's a lot of people online today, and we truly appreciate that. But, for those of you that are new to Syner-G or are joining us for the first time or have just heard us and decided to pay us a visit, who is Syner-G? So, this slide really sums it up fairly concisely. So we are a consulting firm that help emerging and mid-sized biotechs, some large pharmas as well, move idea from IND to approval with few surprises and cleaner execution. We do it across 3 integrated pillars: science, strategy and transformation, and technical operations. Within the science space, this is our CMC core and what this Ask Me Anything series will be focused on. So it spans drug substance, drug product, analytical, and both within the realm of small molecule and biologics and large molecules. And all of our CMC technical side is tied to global regulatory CMC, and we have an incredibly close working relationship with our regulatory CMC partners, and overlays a pragmatic quality approach. We really believe in building phase-appropriate plans that generate appropriate data and write the sections that get filed and defended to allow you guys to move quickly through the drug discovery and development process. Within our strategy and program teams, we recognize that programs don't often fail only in the lab, they fail at hands-off and transitions. So we help teams design operating models that keep science, regulatory, clinical, and finance moving in one direction. We try to do the right sizing and delivery and execution for at the corporate level, with a very specific focus on biotechs and pharma. Lastly is our technical operations and delivery infrastructure. They make the plan run and the plant work. So Syner-G helps biopharma companies scale efficiently and operate with confidence. We provide right-sized technical expertise, operational leadership, and hands-on support to turn strategy into scalable, compliant execution to set up your your manufacturing and commercial success. So as a company, that's who we are. We're highly integrated, highly collaborative, and highly cross-functional. And you're going to see that today as we go through a series. We've got a couple of questions pre-planned to start the conversation, and hopefully you guys as listeners and participants will populate some questions that we can ask of the panel and get those topics discussed. So, one of the things that really makes Syner-G strong is that we consistently deliver because we fuse 3 disciplines that are usually siloed into one accountable team. First, there's the hands-on technical development across all the relevant areas. Second, there's the regulatory CMC leadership that shapes the decision and the data and the reviewer-ready dossiers. And then third, we embed phase-appropriate quality and operations that make programs work smoothly, be compliant, and it's one operating model. And with leaders that have worked together and teams that have worked together and know what works because they've been there and they've done that. So without further, unless there's any questions or comments that I don't see, I'm going to start by asking the panel the first question. So before first in human, what does phase-appropriate CMC actually look like, and where do teams most often overshoot or undershoot? Who would like to field that question?
Yeah, I mean, I can certainly start, from the regulatory perspective, and hopefully, really when we talk about first in human and phase appropriateness, I think a lot of that has to do with like informed decision making and really sort of understanding disease indication, patient population. I'll give a shout out to the target product profile as well that can really sort of help guide on what really needs to be done when, and obviously not doing too much too soon before you get to sort of proof of concept, but really sort of understanding what's actually necessary to get to that phase 1 piece. Maybe analytical development, maybe formulation development, maybe I'll pause there and let my colleagues jump in.
Yeah, and I would agree absolutely with you, Drew. I think that one of the things about phase appropriateness, in thinking about it, is it provides you with the sort of foundation through which you can follow the rest of your development path. So at those early stages, I think it's important to, from a CMC perspective, to understand what the must-haves and what the nice-to-haves are, and really delineate things into those buckets. So when you're talking about like analytical development, for example, and I'm speaking from the purview of large molecules, Ed can certainly jump in from a small molecule perspective, but some of them are very similar. From an analytical perspective, I think what you really want to, ensure is that you are not doing too much too soon. For a couple of reasons. One, you don't need to do that at an early stage. So your analytical methods need to be qualified, but they don't need to be validated. In terms of specifications, you can set your specifications, but you set them in a data-driven fashion, but in a broad fashion to allow you for flexibility. And all of these things allow you to, sort of further develop your program. So if you tighten things, if you validate your analytical methods or you tighten your specifications too early in the process, you've sort of created a situation where there's not flexibility to move. But I'll turn it over to Ed and see if you want to, sort of chime in from the small molecule perspective.
Yeah, I'll chime in, but maybe offer a little bit of the counterpart. I agree
Yeah.
with everything you said as far as not locking yourself in too early. At the same time, there are some places you do need to invest, and that's really in understanding your molecule, or making sure that the molecule you have is staying the same for what you want to do, especially pre-IND phase. And what that often means is making sure you're investing enough in your reference standard characterization, and the maintenance of that reference standard supply. That's a precious supply. That is the linchpin of every analytical result you're going to generate through your GLP tox program and likely on into your phase 1 studies. So making sure you've got that adequately characterized. And then my little bit of a soapbox here is a reference standard is a well-characterized material. It doesn't necessarily need to be an extremely highly purified material.
Yeah.
And so, you don't need to invest a lot of the effort in getting something that's 4 nines pure, but you do need to make sure it's well characterized and you understand what it is, and then you've got it stored in a manner that is going to keep it that way. So being a little bit more conservative with your reference standard characterization very early on, and as you get more data, loosening that up as you move through the development program.
Yeah.
Go ahead, Drew.
No, I was going to say, I think we tend to find, and maybe from my perspective, that there's a lot of overshooting early on, and not necessarily that sort of science-driven and understanding what that product is and what your potential development timeline will look like. We see a lot with early stability programs. We see that a lot with, I think everyone sort of touched on the method validation or method development. And some of these things are, I'm not going to say, we want people to think scientifically, rationally, and risk-based. And a lot of times working with larger, external organizations that have multiple SOPs in place, that there's sort of a battery of tests and a battery of procedures that they just sort of blanket across the board. And you as an individual sponsor that's sort of, looking at your own purse strings and looking at what needs to be done, really needs to take into account that phase appropriateness. And that's why having external experts and folks that have done a ton of different filings throughout the life cycle, can easily jump in on what those nice to haves, must haves, and really what the push point or pain points that some of the agency feedback has been over recent years. Sorry, Ray, stepped on your
No,
toe.
I was going to say, from a bigger level, I think it also varies depending upon the company risk profile, right?
Yeah.
What they're willing to tolerate for risk, and I think that's where having consultants and outside experts kind of weigh in terms of what's I don't want to say industry standard, but best practice, and having the vision that it's a journey. It's not a discrete destination in IND filing, and that you're setting yourself up for years of engagement. So, trying to commit too soon, too quickly, too tightly to a specification or a certain path, you're just likely setting yourself up for failure and more difficult work and additional expenses that you might not normally have encountered if you had kind of the destination in mind, not necessarily the discrete moment of an IND filing. So I don't know.
I think you touched upon one of the places where we've seen a lot of clients struggle is feeling more comfortable in the later stage space and having things well defined and not knowing where they can maybe relax the expectations. And that's what we can bring to those discussions.
Exactly.
Yeah,
So Yeah, Drew, I was going to move on to have you, because you touched on the the TPP. So, let's have a discussion on that, because I think that's like the natural progression of, how and when do you start with a TTP and evolve it into a living QTP that truly drives CQA's control strategy, coherent filing, and kind of without getting buried as a nice to have within the program. Like, how do you make it a useful tool through the drug development process?
Yeah, I mean, I think it's a great question, and it's something that I maybe is one of my sort of passions as like a regulatory professional over the years, knowing that, Q8, Q9, Q10 sort of came out earlier in my career with QBD, the ICH guidance documents, and FDA sort of jumping on not the bandwagon, but sort of in the early 2000s, trying to get industry to think through this target product profile, which, there's a draft guidance out there from 2007, and I think that sort of got a little out of control. But the TPP is a great tool to utilize, as I think prior to the call we're talking about when do you start with it, and I think it's probably at candidate determination. So early on in the development, to have like that key core team really understand what are we driving to and how do we measure success. So if you're not familiar with the TPP, there's a lot of guidance documents out there, but it's big bucket items. So like how do we measure success? What is the nice to have? What is the must have? And where do we need to sort of like align as an organization to like have a go no go decision, and that sort of drives some decision making when you talk about phase appropriateness. Like, are we in a highly competitive area where we think we're going to be from a disease indication or a patient population, and do we need like a really keen formulation or something that's more second generation to initially kick things off in early on in clinical development. But selfishly, as a Reg CMC person, I want the team to think about a TPP so then I can then leverage what FDA, ICH has come up with, is the quality target product profile, and start linking our CMC team to like what is success. So, and we want to make sure that we're aligned with that core team. I have a lot more to say, but I'll pause there because I know I talked a little bit, but yeah.
Yeah, I would say that I like to have the TPP, at least a, sort of a high level TPP in place early on, so that you can really understand the what about the molecule and the program. And then as you transition into, sort of heading toward CMC, I like having that QTPP in place so that, you're starting to get, as you're developing CMC, you're starting to get your CQAs in place. You're starting to understand what the critical process parameters that support those CQAs are, and that's really going to be defined in your QTPP. So I like to do it as early as possible. And again, sort of in a high level fashion, going into a more, sort of formalized fashion as you move through the process.
Yeah. Well, and the thing is, it's a relatively light lift. I mean, the pieces of a target product profile are things that companies are always talking about in terms of what they want it to be, and it really becomes a document then that gets everybody's agreement. And then, as from the CMC side, as we're generating data, it allows us to put those data into context. And and along with the TPP, I also like to have the discussion around the MAP, or the minimally acceptable profile. Right. If I'm thinking about drug product design and anticipated shelf life, for example, or storage conditions for a sterile product, right? You can spend a lot of time solving a formulation problem that maybe where you're able to meet the MAP but not the TPP, and you bring those data in that context and have the discussion at the corporate level and make the decision, hey, for the phase we're at, are we good to go now, or do we need to go ahead and invest the additional work that may delay things a quarter or two to really truly hit the TPP? And those are the important discussions to have, and what that drives just from a corporate decision making standpoint, in addition to making Drew happy.
Right.
Yeah.
Thank you. Yeah.
Yeah, I mean, I'll pull it up from a business risk perspective, because I think the thing that I really appreciate about TPPs at any stage, particularly as they progress with programs, is it gives the entire team a document that you've all agreed to kill a program, And that's a really hard thing to say, and those are really hard decisions to make. But by giving yourself that allowance up front, say this is what we need from a company perspective, a risk perspective, and a consumer perspective and patient perspective to meet our demands. It just helps. It guides all your decisions. It can then guide what work you need to do, what work you don't want to do, and then it makes it a lot easier to bring back to senior management and say, well, here are the reasons why we need to kill this program, or here are the reasons why we can't kill this program because it's meeting all of this. And I think those conversations can be really powerful when the tool's used correctly, outside of just thinking about like how to develop your specs.
Yeah, now I'll just add, when you get sorry, go.
No, go ahead, Drew.
Real quick, I'll just say, as you sort of march through development and you're trying to tell that story when you get to the marketing application stage, be it NDA or BLA or MAA, it tells a really good story. And we talk a lot about like storyboarding and storytelling that, from day one, we've had this concept in our mind of what the TPP what the product is, and then how that relates to our pharmaceutical development activities, and then ultimately tying those attributes of the quality target profile. Because you want to be designing a product that's going to be successful and used in the marketplace, but also tying to product quality with the CQAs. And that's something that happy to talk to any individual on, because it is sort of a passion of mine and a lot of folks about telling that story. And it really helps the reviewer from the CMC perspective that's jumping into an application maybe they've never seen before to understand that story and why certain things were done the way they were done during development. Stacey.
Yeah, I absolutely agree with that. And I'm going to go back to something that Ray said that I think is also really important in an overall program development perspective is the TPP is an opportunity to bring all of the different potentially siloed areas together: regulatory, quality, technical, as well as clinical, because all of them need to have input with regard to that TPP. And so getting everybody then on board early on in terms of the program development, I think is imperative. And then, Drew, what you just said about the CQAs is a particular passion of mine as well. I absolutely agree. It does allow you to sort of create that early narrative, early storyboard of how the development process has occurred. And for regulators or somebody who is not close to the program, getting that high level understanding is important.
Yeah, so I'm going to ask you guys a kind of a surprise question, because I've been thinking about this. Is related to TPP. I know in my experience, some of the most challenging programs have been when companies approach us with essentially a commercial ready phase 3 clinical program, but no TPP, which then results in them trying to retrofit some CMC work that's been done to a TPP that makes sense. How have you guys found that challenging? And if so, how is that either hurt or helped a program at that phase?
Yeah, I mean, I think I'll start, and I think you don't want to pretend to be something that you're not. So if you've gotten to a phase 3 program without sort of, and the clinical data is supportive, there's certain things that we can do to put pen to paper to sort of help fit the paradigm of what agencies tend to expect when they're reviewing a dossier. But I certainly wouldn't spend a lot of time and resources maybe going back to retrofit into a square peg into a round hole. But I'll continue to think and see if I can pull up an additional example. But
yeah, I think if it's a company that's coming in with something that's phase 3 ready, you've got your development story that gets you to that point, and that's where the TPP is really important. Certainly from a CMC standpoint, when we're talking phase 3, I'm starting to really focus on, I'm thinking about the robustness aspects of whatever choices have already been made. And that's the focus that's really going to be, that's going to be the primary focus of, any commercial application, whether it's an NDA or an MAA. And so, making sure that we understand where the pinch points are in the molecule or the drug product process, where the weak points are in those, because there's always a weakest link there. And if I'm going to focus anything back on the TPP, it's going to be looking at those, using that sort of risk assessment approach to identify the weakest points in the formulation or the process or the molecule, understand the risks around that, and what were the choices that led up to where we are there, and how are we going to manage them looking, through phase 3 and commercial.
Yeah. Well said.
Yeah.
Thanks, Ed.
Good job.
Yeah, absolutely. And one of the things I think is challenging in that instance, and I've been a part of, sort of organizations that have gotten to phase 3 without a QTPP in place, is retrospectively then going and trying to generate that documentation. And I think the most challenging thing is to do it in an objective fashion. So start with the data that you have, and like Drew said, don't try and fit, sort of a circle into a square hole, but build off of what you have. And utilize that with regard to, at phase 3, you should have already defined what your CQAs are, what your control strategy is. And so, sort of understanding and taking into account all of that information to build an objective retrospective QTPP is the approach that I would take.
Yep.
Yeah, maybe.
Yeah. So we have our first question from the attendees, and I think it's relevant to this. I'll any of you guys can pick up on this. So it's when to use QBD and build a control strategy. I know I have my personal opinions on this, but I would love to hear your guys' thoughts on when it's appropriate to use QBD.
Yeah, I mean, I'll start. I mean, QBD was sort of this abstract concept or theoretical sort of philosophy back in the early 2000s when it first came out. And, I spend a lot of time, a lot of us within Syner-G have a strong Vertex background, and Vertex was sort of an early pioneer in sort of leveraging QBD to the nth degree. And it was fantastic. And I think the concepts within pharmaceutical development, ICH Q8 and Q9 and Q10, are really sort of best practices now in any sort of drug development activity. And I think there's aspects that are sort of incorporated into the work that's done through the things that we've talked about, including the TPP, including sort of understanding when you want to have certain hard discussions, or those discussions on data generation to support CQAs and critical process parameters. All those things have sort of really become normalized, and I think are part of the process as drug development now. So early on, I mean, you sort of, from that preclinical phase 1, phase 2, through proof of concept, with really sort of ramping up some of those DOEs and type work, around that phase 2, getting ready for phase 3. If you're in a normal development timeline, right? Expedited and other types of timelines kind of throw things out of the water, knowing that CMC tends to be rate limiting.
Yeah, I mean, so I would add, it's never too early to start using QBD, right, in DOE. I think it's an extremely useful tool that can even be used very early on in development as you're developing your processes, because I think it can, if applied correctly, can save you a lot of time further down the development path. It's a great way to get a lot of data quickly, understand points of failure, and really put that into a process that makes sense and is scalable and reproducible. So that's my 2 cents. And again, that comes from my days at Vertex when we were trying to do it. I was there a little bit before Drew in the early 2000s, when we were really trying to put QBD and DOE with everything that we were doing in the lab.
Yeah, I guess my thoughts on this is, as QBD rolled out, one of my initial thoughts was, this really isn't all that different. This is just a framework for the things we were doing already anyway. Right. And the other thing that is, I try to make sure I'm trying to remind people of, is QBD is not DOE. DOE is one element of QBD.
Yeah, QBD.
But QBD is really understanding your product, your process, and the risks associated with it, and having a forward-looking approach to how you're going to justify your control strategy. Yep. And in many cases, that's very straightforward or built upon experience that you have, say, with a particular manufacturing process, and you're leveraging all of that knowledge that you have from other products that you've developed, and you're bringing that in, and you may not need, that may be perfectly adequate to say, we understand this design space, we don't need to do anything more. However, for a particular product going into a particular step of a manufacturing process, yes, if you identify risks there and risks that you don't yet understand, then DOE is potentially your tool to go ahead and develop that understanding. But they are not, QBD is not synonymous with DOE.
Right. Agreed.
Yeah, good answer, Ed. Thank you.
Yeah.
So
I would agree.
Go ahead, Stacey. No.
Sorry, go ahead. No, I would just add, I would agree, setting the stage early as possible with regard to QBD is probably beneficial. I think that, in terms of initiating your control strategy, I would also suggest that as early as possible, but you are going to be limited by the data that you have. So as Ed mentioned, as soon as you get enough data that you can start to leverage to begin to craft your control strategy, I would start to do that, whether it's, sort of during phase 2 or post phase 2 going into phase 3. That being said, when you're talking about accelerated approaches, and I think a lot of the clients that we're dealing with are dealing with an accelerated CMC approach, you need to, having an understanding of and some forethought with regard to how you're going to do your control strategy early on is going to behoove you, because you're going to need to be 2 steps ahead with your CMC, especially if you get good clinical data early on.
Yeah, maybe I'll just add one more thing. Agree with everything that was there, and obviously understanding what's important, what's not important, meaning what's critical, what's not critical, telling that story, and where to sort of focus your energies. And then that's sort of outside of where we are with ICHQ 12 and a couple other things. We're talking about established conditions, where it's a little bit more regimented in the regulatory reporting of certain attributes and process parameters. But really, you in your facility and understanding your product, what do you need to focus on from the critical aspects? And control strategy is not just specs, it's everything that goes into like how best to manage and understand product quality and reproducibility of material, regardless of facility and whatnot. Good. I see we had another question, right?
Yeah, we did just have another question. It kind of rolls in. It's actually a really good bridge, because the next question that I was going to ask you guys is about CDMOs and outsourcing. And I think that's a really good topic, I think, to move into. So how when and how do you lock in your partners, right? The majority of the world is going to an outsourced manufacturing model. So how important is it to plan check transfers, secondary sites? Because I think one of the anybody that sits within CMC, we all never want to be the bottleneck or kind of what a submission's waiting for or a failure. So, within working with the CDMOs, that a lot of our clients, and I'm assuming a lot of people on this call work with, how best to approach that, and what's the philosophy, and it may be different for small molecules versus biologics. And Drew, from a regulatory perspective, what are some ways that you've worked really well with CDMOs? Because obviously there's that transition as well, right? There's a lot of responsibility that clients are placing within CDMOs that impact the regulatory filing. So how do we manage that?
Yeah, I mean, I'll start. Obviously, as we if you think about marketing application getting ready post phase 3, or even before phase 3, understanding what, again, I think Ray, you mentioned risk tolerance, and why do we need to either sort of lock in CDMOs or make additional modifications and add CDMOs or different testing facilities. And one of those things is really to have those individuals, or that CDMO, as part of your marketing application team. They're a partner. You really need to understand, how you think you have designed your process is being implemented at that, particular manufacturing location. And then ultimately, when it comes to regulatory review and of marketing applications, regardless of the jurisdiction, the CDMO and the place of manufacture plays a very important role. What's being done at the facility needs to equate to what is being described in the dossier. Then also you have the added layer of GMP compliance and sort of expectations from that perspective. And I think I would just state that like you really can't underestimate sort of that linkage between the marketing application, GMP readiness, and sort of regulatory and CMC development, if you will. Yeah.
Stacey,
Yeah, I can.
Yeah.
Yeah, I was just going to say I can take it a step back in terms of even, sort of just identifying the CDMO that is going to be appropriate for your program. Because as Drew said, you want them to be your partner. And you want them to be able to walk in lockstep with you as the development of your program moves forward. So, from that perspective, we do a lot of CDMO selection. And one of the things that I like to do is really get an apples to apples sort of comparison of CDMOs and look at a number of different things, including like their capacity, their queues, their regulatory track records, are they a one-stop shop, or can they only do DS, or can they do DP, or do they ship analytical out? Is analytical internal, and if so, what is their strength from that perspective? And then from the perspective of, sort of business terms, right? Especially from the perspective of biologics and cell line master cell banks, I think it's important to understand where your CDMO sits from that perspective. And then a geography perspective. I mean, this has been a conversation that we've had a lot over the past year with regard to, sort of geopolitical environments changing and where you want to do your manufacturing when it comes to, sort of moving your product across the world. That's something that's definitely being taken into consideration. And so what I like to do is sort of have a tool that levels the playing field for all of the CDMOs so that you can look at a direct apples to apples comparison of all of them. Because I think it's really important for you to, sort of select early on that CDMO that potentially is going to be a CDMO that can help and work with you throughout the entirety of your program, all the way from, early stage through to marketing authorization. I'll leave it at that. I have other things, other things about tech transfer that I can talk about.
Yeah. And I think that's where, Stacey, I think your experience is colored more by, operating in the biologic space. I think we've got a little bit more flexibility in the small molecule space as far as ability to change CDMOs through the development life cycle. And that's where, the decisions really depend on what the objectives are of our clients. Are they looking to get through a phase 2 proof of concept study and hand it off to another development partner? Do you have a high risk or a low risk, formulation, right? If you've got a simple solid oral dosage form of a BCS class 1 compound, you've got a lot of flexibility because your risks are much easier to manage than if you've got poorly soluble compound enabled formulation and, on top of that, potentially a large market where now what you need for commercialization is a much different scale than what you might have needed to get through certainly the phase 2 study and maybe even a phase 3 study. So it's, a lot, we've all come through the science, and, that's where I tend to go back. And it's the better you understand your product and the demands that you need to meet, the better you can make decisions about what type of CDMO to partner with when.
Yeah. I mean, to that point, I think it comes a lot down to, analytical methods, particularly in the small molecule, in terms of the comfort level of changing kind of last minute. Because it's really about understanding the molecule, understanding the science, and what the risks are with any change or any partnership. So I think the other thing that I will bring up that hasn't really been talked about at all, and you guys feel free to comment, is the idea that quality is a culture and a mindset, not a list of SOPs. And I think, particularly for CDMOs, trying to understand that, going to failures or 43s or citing, CRLs, it's because, in my opinion, and this is my opinion only, but if a manufacturer thinks that everything is contained within SOP, and it's not the mindset and the culture coming from the most senior levels that quality matters, and you're going to fail, right? Because what ends up happening is willful disobedience of SOPs because people think they're doing it better, quicker, it makes more sense. They're not capturing changes that should have been captured because they don't recognize the impact on the overall program. I think that's where manufacturers get in trouble, is when they haven't fostered and built that culture where, quality and putting the patient first and safety is of utmost importance.
Yeah, I mean, I'll add and just sort of state that that critical thinking, scientific thinking applies to all realms within pharmaceutical development, including the quality organizations, and having appropriate SOPs that are in place to sort of garner that facilitation of knowledge transfer. I think maybe just like really not underestimating the not underestimating the time and energy it takes to become a good partner, you as a sponsor with the CDMO, and really working with them. And I think those who've been in the industry for a long time have really sort of understood that CMC could be rate limiting, facility inspections could be rate limiting, that ultimately could lead to CRLs, and trying to bang that drum to get upper management to understand that time and resources need to be spent on that. There's always been, and again, this is my opinion and my opinion only, and I think maybe some other folks share that, if you have fantastic clinical, and maybe this is maybe more focused on large molecules. If you have fantastic clinical data, this concept of CMC reviewers are going to roll over for clinical reviewers. If that data is so fantastic, it's an unmet medical need, we need to get to market. What we're finding is, like, especially in complex dosage forms and multi-use facilities, that risk benefit to the agency to allow approval of something that could be suspect on the quality or CMC development side isn't holding true. And they're really putting the feet to the fire of those organizations to sort of get their act together, regardless of the clinical data that's been out there. Now there's a lot of nuance. There's a lot of specifics that we can get into. I think small molecules could be slightly different, where there is some maybe more, I'm not going to call it flexibility, but the ability to sort of meet requirements in different manners, where it doesn't hold true for the large molecule space.
Yep.
And I'll let others sort of chime in.
Yeah, I've had direct experience with regard to that, Drew, where upper management in a company believes that they have wonderful clinical data and do have wonderful clinical data. The FDA acknowledges that, provided a package to the FDA, Module 3 package that was not sufficient. And, the upper management in that scenario thought that they would be ushered through without any issue. And in reality, the FDA was still concerned about, appropriate control in terms of manufacturing, and control strategies that were, sort of underdeveloped. So it ended up taking this company an extra 2 years to overcome a CRL that they got, that if in reality, they had taken a pause and maybe spent a few more months to address the issues, make sure that the QTPP was appropriate, and that all of the CQAs and control strategy really followed, sort of, and supported that QTPP, I think they would have been in a much different scenario in terms of approval. I mean, it ultimately got approved. It just took longer.
Yep. No, great conversation so far. There's a couple of questions in the chat. I'm going to purposely not answer those because they're a little bit specific if we have time, but I do want to get to one of our last questions because I think it applies pretty broadly. So for those people in the chat that have questions, we will follow up directly with you and open some conversations because some of them are pretty specific. So, I think everybody's aware that budgets are really tight right now. So, I want each one of you guys to answer this because I think there'll be different answers. So what are the top 3 CMC investments that you'll refuse to cut early on in development? Let's say first in human, because cheap now is expensive later, and they're always going to come back to bite you. And how or if do you use structured risk assessments to justify the spend? Stacey, I'm going to start with you. Sorry.
Okay. Yeah, no. I think that, one of the things that stands out from my perspective that I would never sort of choose to wait until later on is the master cell bank development. And I wouldn't, there are certain things that I would sort of make sure that I understand with regard to clonality and, sort of sequence and identity and evaluations from that perspective. I think you can push, some of the MCB stability for sure, but that early stage sort of understanding of your master cell bank is imperative, especially for large molecules. That's the foundation of your product. And a lot of companies that are, sort of in that early phase are trying to make decisions to move forward with things that might not be, from an MCB perspective, might not be what they want later down the road. And changing that later down the road is going to be a very expensive, from a monetary perspective, as well as from a time perspective. So I know that you wanted a couple things, but I think that would
let's
be the
go
thing that I
one
would
of
harp
each now and then
upon.
circle back.
Yeah.
So Drew, how about
Yeah.
you from a regulatory perspective?
I mean, I would say not really, how do I say this? Having a data-driven understanding of CMC changes that could potentially impact pivotal clinical results versus your commercialization. So right, in the small molecule world, we make a lot of changes out through development, but really understanding the impact of making a change during pivotal clinical, maybe prior to registration and then commercialization, and building that bridge, because it all comes out in the wash when you're telling your story. And if you don't have the data that to support why things wouldn't impact the safety or efficacy of the clinical readout, puts a lot of sort of a lot of pressure on the team to sort of start generating that data during the course of NDA or marketing application review. That should be part of your story elsewise. That's
Yeah.
Okay.
So
And how
Drew,
about you?
I just, oh, sorry.
No,
sorry.
You want to say something, Stacy? Go ahead.
Yeah, go ahead, Stacey. Sorry.
I did, because comparability is another area that's like near and dear to my heart, and especially in the large molecule space, when you are making these changes, it's imperative to do it in a prospective fashion, such that you have a protocol that is driving this, and that these reports are clear. You're often going to be using that comparability and leveraging that information to, to justify why you don't need an additional clinical study. The other aspect of it is, for large molecules, it's not an analytical comparability largely.
Right.
You're looking at process comparability as well as analytical comparability. And that is a huge task. It's not a trivial matter. So really prospectively thinking about that is critical. Sorry, I feel very passionate about that, Ed. Go on.
Yeah, no.
But I knew that needed to follow on Drew's comment. So, and this is a place where the small molecule world is different. And Ray, you asked the question in terms of earlier phase, so I'm going to kind of skew my answer that way. But I come back to the two main things we're really most worried about, which is impurities control on the CMC side, making sure we don't get things in there in the product that we don't want there, and then reproducibility of exposure or performance. And so investing early and making sure your impurities method is robust, that you understand the extent to which the stability indicating to minimize surprises, assuming you continue down the pathway. And then particularly for the more challenging molecules, what are your options for ensuring robust and consistent exposure? And to that end, understanding what the physiological factors are that might impact that, because that's still a place where we've got handles that we can pull from a CMC side to deal with that if we understand them. So a good partnership with your DMPK colleagues early on to, again, think about those risks, understand them, make sure you're interrogating with them the early PK data to understand where those risks might be, so that we can make good decisions on the CMC side about designing some robustness into the formulation where it's needed.
Yeah. The one thing that I would add that none of you guys touched on that I do think is really important, particularly for early stage programs, is thinking with a global mindset. A lot of our clients and people we've worked with in the past come and they're only focused on going to Australia or going to the US, and when you approach it with that mindset, you write and you have CMC justifications in a specific manner, but it makes it much more difficult then to back up and use it globally, which 95% of the time people want to do. So I think, adding that little bit up front to say material is going to be QP release. We're going to think about, how we're setting up our stability, how we're setting up our specifications to apply globally rather than regionally, is really important, particularly for early stage programs that want to move quick. Because the number of clients that I've seen that come in, we're only going to Australia with that material, and then 3 months later, their clinical team's trying to file that same material in, the UK, the US, Canada, and it just leads to a lot of complications and typically delays. So, Stacey, Drew, I don't know what your thoughts are on that, but that's one of my things that I always push clients to think about, that it's worth investing sooner rather than later in that mindset.
Yeah, definitely agree. I think understanding where the material is being made, where it needs to go, how it's being made, and I think some of the things that we've run across in the last 10 years or so is the ability to import drug into the US without an IND, right? So you have to have the IND active, but if your drug product manufacturer is in the US and you need to generate data, there's some, I'm not gonna say workarounds, but there's that thinking that needs to be made to sort of understand what the long-term development or even short-term development priorities are. So yeah.
Okay, so we've got about 9 minutes left. I'm going to take one of the questions from the attendees. So, how do you build flexibility into CMC development and control strategy for drug device combination products where it is common to change drug delivery device between different clinical stages and from clinical stage to commercial stage? So, I'm going to narrow this down a little bit and say, injectable. Let's just take an injectable because often they go from vial to pre-filled syringe to auto injector. So how do we think about and advise clients in a path like that? And it doesn't have to be specific, but I just want to narrow it down for you guys a little bit.
Yeah, I mean, I'll start, and I think if you're specifically looking at flexibility, sort of understanding what you're dosing, or understanding the product to be dosed, and then how it's delivered, and then obviously the control strategy that is sort of in place is going to be modified based off of the delivery mechanism, or how it's being delivered, and yeah. Sorry, I lost my train of thought here. I started reading the question again. Again, I think it's sort of like, again, going back to like a TPP, or understanding like what the sort of basic needs are, understanding that eventually if you know that you need to be in the marketplace with one particular method of delivery, like for patient convenience or patient compliance, to sort of build that in as you sort of are moving through development, and having an understanding of how that product behaves and how that product could potentially behave in different delivery forms. So pre-filled syringe, vial, auto injector, could be different depending on physical chemical attributes of the product that you're going to be delivering.
So Drew, I hear you saying the more you understand your product, the better you can manage those changes.
Thank you.
Well, go ahead, Stacey.
Well, and understanding what the long view of the program is, right? Oftentimes you don't have that in advance. So if you can have that understanding, and if it's driven by the TPP or the QTPP, that's great. If not, but having that long view is important to understanding where you can then build in flexibility. If you, are starting with an IV program where your concentration is 10 mg per mil, that's a different thing than going to a sub-Q formulation that's going to be 150 mg per mil, right? Where you're going to have a lot of, sort of robust changes in formulation that would support those different presentations. So I think that, it's a hard question to answer definitively, but I think there are a number of points early on that you can think about that would set you up strategically to, sort of have an appropriate flexible control strategy or program moving forward.
Yeah, and I think the key thing with that is, if we've got an understanding of what the ultimate goals are, what those changes might be, it becomes easier to design studies earlier on to screen some of those things and identify where the difficult problems are going to be to solve. So if I think about changing product contact surfaces and potential for active ingredient absorption to different materials, and we know that we want to move, say, from an IV bag to a glass or a plastic, primary package. If you know about that sooner, you can screen them while they're not on critical path, and have a chance to know whether or not it's likely to be an easy path forward, or it's going to require more investment. If you get the data that says it's going to be a difficult, more difficult to solve, you then have the information to build the case that, hey, we need to fund this work a little bit more. We need to de-risk this, and here are the studies we think we need to do, or accept that, if we're going to put that off, there's now an increased risk of a delay in moving to the next phase or that next presentation.
Yeah.
Go ahead.
No, I was going to say, I think this is where the experience of a consultant or somebody with broad experience can really come into play, because we have the luxury of seeing a lot of times what the end looks like and how people got there, because we've worked through similar compounds or similar indications or similar molecules. So I think, unfortunately, CMC tends to be more reactive than proactive. And I think taking a proactive approach to drug device combos early on, knowing the market, I'll be, really specific for GLP-1s, right? Like, everybody now is going to a longer acting, injectable. So, if you're starting your clinical program with solution in a vial and you're injecting it, you're probably not going to end there, right? Like, that's no longer commercially viable. So, even if your commercial team hasn't told you that's what's a presentation, from a CMC perspective, you should be thinking about how you're going to achieve that, and, factoring that into your whole development program. So
yeah, I was just going to add the fact that, like, it's really cross-functional, and any sort of modification to a delivery system, like, you need to look at sort of clinical implications, clinical efficacy studies, and obviously directions for human use, the risk, human factor studies that sort of all leads into modification of, like, the delivery method. Typically warrants, a lot of discussion and a lot of, further decision making. So that's why the TPP is a great place to have those.
So I want to be really respectful of everybody's time, because I really appreciate everybody participating in this. I want to thank Stacy, Drew, and Ed for being fantastic participants. And any final thoughts to leave the participants with before they go off to their next meeting or scramble to grab some lunch?
I'll say, happy to have any conversations with anyone on any particular topic, for sure. I saw there was a couple of questions that are sort of near and dear to my heart that are in that we didn't get addressed to, so we can certainly take that offline and help out that individual. So yeah, but yeah, think with the end in mind, maybe.
Yeah.
I think you just captured it really well right there, Drew. Is the further down the road you can see, the better prepared you are for what bumps there might be in the road.
Yep, I would agree with both of those, and I'm happy to hop on a call with anyone who has any further questions, either from the Q&A chat box or in general.
So with that, I just want to thank everybody, and we'll wrap up, and look forward to speaking with people. Hopefully this has prompted some conversations that you might want to have. And very honestly, if there's other topics that you think would make good Ask Me Anything series, please send those to myself, and we would love to put those on for a broader group. We really appreciate doing these and getting in front of people. So thank you very much.
Thanks, everyone.